wine fault remediations, and I'm Andrea Botesato, Assistant Professor and Enology Extension Specialist at Texas A&M, Texas AgriLife Extension Services, and our presenter today is Luke Holcomb from Scott Labs. Welcome, Luke. Thank you for being with us today, and please start your presentation.
All right. Thank you. Thank you, everyone, for tuning in. This is, I guess, part two of a three-part series. The last one we talked about what defects there are and where they come from and that sort of thing. Today we're going to talk about remediating wine defects, and this is a huge topic. I'm going to focus in on a few particular defects that are very common and how to work with them. If you guys have questions, I think we'll address them at the end. And then the next part of the series will be prevention, because prevention seems to be and is most likely the best option. If you can make good sound quality wine to begin with, it helps out a lot. So let's cruise in and get going here. So, this is kind of a recap of the last presentation on what could be a defect. And some of these, depending on your philosophy of winemaking, may actually be just an attribute of your style. I saw something on Facebook the other day where a winemaker said, I'm making rosé today, and there's going to be no filtration and no fining, et cetera, et cetera. Made that declaration on the front end. There was juice at that point. It's going to be a wild ferment as well. So clearly, his philosophy sounded like it drove his winemaking more than maybe perhaps wine quality. So when I look at, and I think most consumers will look at defects as anything that negatively affects color, appearance, aroma, mouthfeel, balance, or stability in the bottle. And so, here we have color. This could be the development of brown hues in the wine. It could also be pinking in the bottle, or also can happen in cellar. That's typically with white wines. The appearance could be, a haze to the wine, excess turbidity, or precipitate in the bottle. And those can exist the same time in the same wine. So you can have a haze and turbidity, and you're trying to address the turbidity, but you also have haze at the same time. So you, let's say you filter it, but you haven't taken care of the protein stability issue, which is causing a haze. So sometimes these things are not necessarily mutually exclusive. Moving on from there, aroma, you can have a lot of different kinds of sulfur compounds. I think we're all intimately familiar with those, and we're going to address those because those seem to be very common. Also, the abbreviations of 4EP and 4EG, which is 4-ethylphenol and 4-ethylglycol, which are produced in varying concentrations by Brettanomyces. And then you would have cork taint, which is primarily trichloroanisole or TCA, although if you want to keep yourself up at night, there's also other halogenated anisole from other sources. Bromine can also form a halogenated anisole, and it smells exactly like cork taint, but it's not necessarily from cork. So, as a cork salesman, I get a little cagey when somebody says cork taint because there's a lot of other things that it could be. Moving on, VA, volatile acidity. I think we're most likely familiar with that. Acetaldehyde, which comes from oxidation. Mouthfeel and balance. So this is kind of a difficult topic because it refers to more maybe a perception or flavor matrix rather than a direct fault. So this could be low or high acidity. Personally, a lot of the Pinot Noirs that come from California and the West Coast, for example, I'm not a big fan of because they are very flabby, for example. So that could be a defect, in my opinion, but it is maybe a stylistic decision on the winemaker's part. So it's not a defect in their perception. Mousy Taint, we'll get into that. That is probably the one that disgusts me the most. And we'll get into how that happens and how to fix it. Bitterness and astringency, again, this is kind of out of balance. So we can talk about how to fix those as well. And then also I put ethanol there. So some consumers are sensitive to higher alcohol levels, and especially in Texas, sometimes you get elevated alcohol levels, and it can kind of overwhelm the matrix and kind of be a domineering sensory attribute. Then you have stability, which can be caused by high pH or inadequate SO2, oxidation, contamination. It can exhibit itself as Mousy Taint or 4EP, or you could see turbidity, also effervescence. And then there are other things like legal limits of, let's say, copper levels. You could have label issues where your label is 13.5 and you have a wine that's 15.7. That's a defect in my opinion. And then compliance, and that's another topic. And I don't have enough time to talk about that today, and nor do I want to because it's not my favorite topic. So, moving on. So the first step in fixing a defect, you've got to identify it. I get phone calls on a regular basis, and somebody will say, "Hey, my wine's really funky," and unfortunately, there's not a standard sensory description for funky. It's different for every person. So, I think we as winemakers need to get our palates trained so that when we smell or taste something, we know specifically what it is, so that we can then treat it. So, if you have a funk and you don't know exactly what it is, one thing you can do is phone a friend, right? Ask other winemakers for help. I know sometimes that can be daunting because maybe some pride attached there, but I think it's a good thing to ask your friends to stick their nose in it and taste it and see if they can help you identify it. You can also consult with your suppliers, such as myself. I would throw in there also your educational partners as well. They a lot of times have they can either analyze for it in the lab or do sensory analysis and help you identify it. You can also send samples to folks like us, ETS, etc., and they can not only do sensory screens on it, but then also they can do like a wine quality improvement test on it, where they take the products that they have and treat the wine and give you a best recommendation based on their experience. They'll do the bench trials for you. The costs of that depends on who you use and what the problem is. Also, it's important to run and track your analysis over time. So, especially with things like SO2, you really shouldn't look at it as a snapshot at a point in time. You should correlate it over the lifespan of the wine so that you can identify if you have problems. If you have recurring depletion of SO2 over time and you keep making big adds and they keep binding up and your SO2 drops, that's a pretty reliable indicator that you either have way too much oxidation, or you've got unwanted microbial growth. Could be Saccharomyces, it could be Brett, it could be a lot of different things. So if you see that, that's when you need to start addressing the problem. And obviously, once identified, you have a remediation plan to be developed. We'll get into that. So, what do we have in our toolbox to do this? Well, we actually have a lot of things. We have a lot more things today than we did 30 years ago or 100 years ago. It's kind of interesting if you get a chance to look at writings and texts from yesteryear. A lot of people used lees and kept lees around and moved it around and fined with lees because that's really all they had available to them. We live in the golden age of winemaking, and it seems like every few months there's a new product on the market. So there's some really powerful tools out there. But here's a list. We'll get into each one of these. These are the things that we have at our disposal. So finding agents, we have settling agents which help to settle particulate as well as microbial populations in preparation for fining or racking. So one of the primary devices that we can use in controlling microbial populations is settling, clarification, filtration, those sort of things. And what we're going for is literally the reduction of the population, sequestering of the population, and getting that wine clear and away from those populations. And there's a number of ways to do it. For example, one of the things that I like to do and share with people is, floor yeast, candida, which creates a lot of acetaldehyde, is the microbial population responsible for making sherry. This happens on the very top of the wine and creates a kind of a oily film, white-looking growth on the top of the wine. One of the best things to do is to literally get a shop vac. I would say get a wet vac and use it to actually suck the microbial population, that film, off of the tank. You can skim it with like cheesecloth or a straining basket. So what you're doing is you're literally removing the microorganisms. You can also rack from the bottom and filter. That works too. There's a lot of different ways to deal with that, but racking and filtering, is one more movement of the wine that you're going to pick up oxidation, and using a wet vac or a skimmer or something like that really helps removing those populations. Anyways, moving on. You also have reactive fining agents. So these will bind, and then there's a few different methods that this happens, depending on the fining agent and depending upon the compound that you're actually working with. And this will help to form complexes and allow you to improve the wine that way. And so fining agents I refer to as subtractive, right? So you're pulling something out. So the other method we'll get into is additive. So you just add something to the wine and doesn't require filtering and racking afterwards. We'll get into that a little bit later. And sometimes, depending on the defect, a combination of agents will be necessary to fully fix the problem. So what impacts the efficacy of these fining aids? You need to do proper lab trials, and we'll get into how to do that. Also, the quality of material. I think it's important to purchase these fining aids from a reputable enological supplier. So, as in the case of bentonite, for example, the good suppliers out there are doing a lot of quality control to analyze for contamination from extraneous microbial populations. They have certificate of analysis. We're also looking at the metal contents, which is important with bentonite. And so suppliers, winemaking suppliers, know the eccentricities of winemaking and are bringing products to bear based on their knowledge of that. Whereas, your local chemical supplier may not have a very good understanding and may not be doing the quality control steps necessary. So you want to get good quality material. Also, the method of preparation of these finding agents is very specific, and there's a reason why we have all of these specific requirements on how they're handled and what temperature of water is used, how much water is used, how much time it takes, and so on and so forth. This greatly impacts the efficacy of the finding aid. Also, the method of addition. So, if you just take your bucket of bentonite and just pour it in over the top without mixing the tank or something like that, you're not going to get good dispersion of the finding aid. So, the method of addition is important. A really good way to do it is using a venturi, which you pump the wine through a venturi and it will pull in the finding aid through a port on the venturi. And it really, you can dial in the distribution very well and get very good dispersion and contact that way. The concentration also is important. So, what we mean there is 10 grams per hectoliter, 20 grams per hectoliter, 30 grams per hectoliter, on and on and on. So sometimes at 10, you won't get anything. 20 doesn't do it, but then all of a sudden, magically at 27 grams per hectoliter is the right number. So this is where bench trials come into play. pH. So let's take the same finding aid and apply it to 3 different wines that are, for sake of example, everything is exactly the same except the pH. You can have different efficacies with the finding agents. So that's what you did last year on your Blanc de Bois at 3.4. It might not work exactly the same. Let's say this year it's 3.7. So that's just for example. So that's why bench trials are important. Metal content. So the metal content of your wine is going to affect a lot of the chemistry that occurs when you add a finding agent. So this, again, I'm speaking to bench trials and the importance of them. Temperature, generally speaking, reactions occur quicker at higher temperatures versus slower at lower temperatures. So, when you do your bench trials, you want to look at trying to mimic what's happening in the cellar. So if your cellar is 55 degrees and you do your bench trial at 72 degrees, there can be some difference in reaction there. Also, the age of the wine. Younger wines or even juice is bounces back better from finding aids than to older wines. Buffering capacity just isn't there. And that's a complicated topic on why that occurs. A lot of it has to do, in my opinion, with oxidation level, and then also the formation of colloidal complexes that occur over time. And then also, there is a law of diminishing return. So if you've already fined at a certain dose rate with finding aid, a follow-up ad may not be as effective because there's just not enough binding sites to react with the finding aid. So these are some of the things that can impact efficacy. Here's a quick slide. I'm going to blow through this. I don't want to spend too much time here, but this is a comparison of the different finding aids, and this is not all inclusive, but it gives you an idea of how to choose a finding aid appropriately. Interestingly enough, a lot of these are animal derived. So if you're trying to make vegan wine for all 13 vegan wine consumers out there, you don't want to use much of these products, isinglass, albumin, casein, gelatin, and on and on. So if you have questions about finding aids and which ones to use as a substitute for an animal product, I would suggest contacting your supplier and asking them that question. Enzymes. So this is a fascinating area of innovation that's occurring. You can really do some pretty incredible things with enzymes. So you can, for example, you can enhance filterability. So if you have fruit that came in that was pretty moldy, there's a good chance that you have the presence of glucans, which is going to make filtration very difficult, if not impossible. And so how is this a defect, you say? Well, if you can't filter it, there's a good chance you can't control the microbial populations, and therefore you can have a defect occur. So you can use something like a filterability enzyme to break down those glucans and improve filterability and the processing of the wine thereof. Other things like beta glucanases, this is something you can use to change the matrix. So beta glucanases, glucosidases, they will cleave some aromatic precursors from other compounds, and it will make them volatile where before they were not. So let's say you have a little H2S problem in maybe the white wine. You can look at using a beta glucosidase to liberate the bound terpenes and improve the aromatic quality that way. So this is a way to maybe not fix the H2S problem, but to change its perceptibility by releasing volatiles that were otherwise non-volatile. So that's an example of how enzymes can be used for remediating a defect. And again, this is a complex topic, so if you have something specific, contact your supplier, and we can work through that. So moving on, here's the bench trials, and I like to put exclamation points on things when I'm really trying to emphasize them. Maybe I should probably put things in all caps so I'm yelling off of the slide there. But so what you need is a well-equipped lab, a good mixing plate with stir bars, assortment of pipettes, including micropipettes. And one of the other things that's not used a lot in the winemaking industry, but it is available and it's used a lot in some pretty advanced laboratories, is a macro pipette. So same thing like a micropipette, but it does 5 mil or 10 mil or 20 mil, and those are really handy, especially for repeatability in your analysis. And if you're interested in that, you can shoot me an email and I can send you some links on where to get those things. So you can get away from mouth pipetting, for example. Also, appropriate sample containers and closures. So good graduated cylinders and beakers and sample jars. If you're looking at hazes, I would recommend glass as opposed to plastic, so you can get the maximum amount of light transmission, and you're not the color potential of the plastic is not clouding your perception. Also, a good scale or a combination of scales. So especially with bench trials, you're dealing with very small amounts of finding aid, for example, or tannins. And so you're going to need the ability to weigh out small amounts. You can also do serial dilutions, but this can take a lot of time. So that scale I showed there is a very economical scale. That's probably $80. If you wanted to get a little bit more precise, you're starting to talk $300 to $500. I've used scales that have literally a cage around them, so the air doesn't affect the weight. So if somebody opens the door, there's enough sensitivity that the scale will actually register the wind. So you can spend thousands of dollars on scale. I don't think that's necessary for the most part. Also, a nephelometer. So a nephelometer will give you a reading in NTUs, which is our nephelometric turbidity units. And so you'll see this used a lot when you're talking filtration. You know, at what NTU level do you, what pad or what filter do you need to use? It's important if you're using cross flows as well, because you can't go to a cross flow with really high NTUs. You got to go do quite a bit of pre-filtration beforehand. It's also good, like let's say you have you know, a microbial bloom, and you've been checking your turbidity, it's been very low, and then all of a sudden it spikes. You know, it's an indication of something going on. And so essentially, the NTUs are the measure of how much clarity and how much light transmission comes through the liquid. So it's a very, very powerful thing to use. It's very common when doing bentonite finding trials to determine at what level of bentonite is necessary to achieve protein stability. Also, spectrophotometer. You know, these used to be cost prohibitive back in the day. I think it's gotten a lot better. You know, $2,500, $2,800, which is, I mean, it's a sizable amount, but what you can do with the spectrophotometer is pretty powerful. You can analyze for basically anything that can be degraded by an enzyme. So acetic acid, malic acid, tartaric acid, glucose, fructose, and on and on and on. So I recommend looking into investing in a good spectrophotometer and some good reagent kits to help you ascertain things there in your lab. So real quick, here's a good little slide, and this presentation is going to be in a PDF and distributed if you guys would like. So you don't necessarily have to try to do screen capture or take a picture of this with your phone, so it will be available. So we're going to move on from that. So some bench trials may take a few days to a few weeks, depending on what you're trying to go after. So look at the technical data sheet for the product itself, and it will tell you, you know, how long you need to hold the product to look at efficacy. You know, enzymes can be 24 to 72 hours. Something like a copper finding trial can be done in about 30 minutes. So it depends on what you're working with. You want to try to emulate the cellar conditions. I said that earlier. Use fresh finding aids. You don't want to use expired product because it could have, for example, absorbed a lot of moisture, and it's not what it once was and won't have the same efficacy. And some finding aids require specific preparation, not only when you use them in the cellar, but also on the bench trial. So follow the technical data sheets that your supplier will give you. Sometimes, you know, if you're trying a few different finding aids, for example, or tannins, you might be trying 5 or 6 different ones, and you find that one works pretty well. Well, then you take that one and then use it in different dose rates to identify exactly what dose rate gives you the desired result. And typically, you're gonna try to look for the lowest amount needed to achieve that. Tannin trials, for example, you know, you might find out of, like, in our line of products, we have about 13 tannins, and you know, that's a pretty big bench trial. So you would go through and dose with all 13, find the ones that you like, pull those out, then you start dialing in the dose from there, and you might actually start using them in combination. So bench trials can be tedious, but I think they're very important. So additionally, I wanted to make a note in the following slides. I will have some product recommendations, and I'm not attempting to do a hard product push here. I'm just giving examples. You know, I've sat through many of these presentations in the past from other suppliers, and it's basically a walkthrough of their catalog. And that's not what I'm attempting to do here. I want this to be educational. If you do want product recommendations, please let me know. I'd be happy to do that. So I just want to mention that. So volatile sulfur compounds. I think that this is very common, and it can really take a wine that was otherwise quite nice and make it very objectionable. So on the left-hand side, you have various volatile sulfur compounds that are typically referred to as defects. So hydrogen sulfide, we're all familiar with that. It's that rotten egg smell. It's very potent at very, very low levels. So it can be a big problem. Ethyl and methyl mercaptan, so a little more cabbagey, onion, rubber. I had a Sancerre the other day at a master som tasting that smelled like it had a tire dissolved in the wine. It was absolutely terrible, which makes me question why a master som would put that wine out. But otherwise, it can happen. Dimethyl disulfide, I get more like stewed onion, stewed cabbage. And then diethyl disulfide is kind of a burnt rubber, garlic aroma. And these things, the next slide will give you my personal theory on how these things happen and how they form. I mean, we know that H2S is produced by yeast. We know that the mercaptans can also be produced by yeast, but also through chemical reactions. And then I believe that your redox potential or your level of oxidation can move these compounds around into their various forms. And I'll speak a little bit more about that. But your volatile sulfur compound detection trial is shown on the right. I got this from Bradley Beam, Illinois. You do not want to taste these samples. These are for smell. Cadmium and copper are heavy metals, so we do not want to drink these. So if you're setting up samples in the lab, you need to have some control over who gets to come in and who has access to this stuff, because we don't want to cause any harm. So the procedures there on the right on what you're looking at, essentially, the cadmium will bind up specifically with H2S, whereas copper sulfate is a little bit more broad spectrum. And so if the cadmium sulfate takes care of the problem, then it's H2S. If it doesn't take care of the problem, it's more likely mercaptans or disulfides. Then the ascorbic acid, what it does is it changes the redox potential. It's an antioxidant, and it will push the disulfides back into the mercaptan form, where they will then react with the copper sulfate. So that's what's going on there. Let's move on from there. If you have any questions about this, my information will be at the end of the presentation. You're welcome to call me or email me, and we can discuss even more. So copper sulfate, you can't use cadmium. It's illegal to use. It's good for identifying what's going on. Copper sulfate can be used, but there are legal limits of copper. It also can be pretty aggressive, and because it's a heavy metal, it will drive oxidative reactions. So you don't want to use it willy-nilly. You want to be very focused, careful in how you add copper, if you choose to add copper. I had an interesting seminar that I attended years ago. It was in California. He was a guy that made a lot of barrel-aged Chardonnay, and he was vitriolically against using copper sulfate. But he said if he did have a problem with H2S, he would use a brass batonage stick. So I don't understand the continuity of thinking there, but it kind of made me chuckle, because he whipped this big copper thing out and said, this is what I use when I have a problem. But he would not use copper sulfate. So interesting. So perhaps one of the other methods you can try is using a lighter approach, which is this is an additive approach. So the SIY stands for specific inactivated yeast. And products that we have in our portfolio are something like Regulus or Noblesse that can be used to treat like H2S and these volatile sulfur compounds. And has a few different modes of action, but it's a much less aggressive form of dealing with these sulfur compound problems, as opposed to just using copper. Finishing tannins. This is really cool. There's a ton of them out there. Some of them are more structure, tannin, mouthfeel type of products. Some of them do affect the nose, and so you can use these maybe in combination to build some structure and change the perception of these volatile sulfur compounds. And then, it's very commonly encouraged to do splash racking with volatile sulfur compounds and specifically H2S. And this is a little diagram or equation that I have there to show what happens when you splash rack. Not only do you oxidize your wine, which oxidation is, according to ETS, the #1 defect, and most people have a problem with it, and it is maybe my purpose in life to do everything I can to help winemakers mitigate oxidation and adjust that. But if you splash rack, you are increasing your dissolved oxygen significantly, and also that oxygen is sharing electrons and moving the H2S into literally heavier forms of volatile sulfur compounds. And so you can take a small problem with H2S and push it all the way over into the disulfide form, which you have a much worse problem with. And people say, well, no splash racking works. Well, I would say that it generally does for a while, and then through the aging process, and the wine becomes what they call reduced, and it loses those electrons. The dissolved oxygen goes down. The perception comes back of the volatile sulfur compounds. So this is what happens. You have a beautiful wine that is just lovely in the bottling tank. There's no sulfur problems, and you put it in the bottle, and then 6 months later, it's very reductive, and that is exactly what I'm talking about. And this is why we decant, because we add more oxygen and we move those compounds to different forms. People say that these chemicals, when you splash rack them, you violently stir them, they somehow develop wings and fly out of the wine. I don't think that is what is occurring. So I'm militant on recommending against splash racking. So that's my personal dogma. So Mousy Taint. This is, I said earlier, probably one that reviles me the most. And I'll explain a little bit how it works. So it's very common in high pH wines that have improper SO2 usage.
Hello.
Obviously, when you're dealing with some of the pHs in Texas
I'm good,
around
thank you.
4,
From where?
You don't have a lot of efficacy with your SO2, so this is a problem. We'll talk about a little bit how to work with that later. But typically, you're going to have high pH, improper SO2, excess oxidation, and growth of lactic acid bacteria, specifically lactobacillus, but sometimes Enococcus, and then also could be in combination with Brett. I've found that we like to look at things like, oh, I just have Brett, or I just have lacto, or I just have Pediococcus. And the reality is, you probably have a little bit of everything, because the same conditions that favor the growth of Brett favor the growth of these other spoilage microorganisms. So, you need to have kind of a multilateral strategy with dealing with spoilage microorganisms. So both lactic acid bacteria and Brettanomyces can produce Mousy Taint, and you can't perceive it by smell. You can only perceive it once the wine leaves the palate, whether you expectorate or you swallow. Then the saliva in your mouth raises the pH of the residual wine, and then the mousiness becomes perceptible. Give me a second, I'm going to step away and close the window real quick. So remediation of Mousy Taint. Typically, you can't filter it out. When I was in California, we had an issue with this, and we tried to have BA filtration come out and filter the compound out. That did not work. So classically, the addition of SO2, like 100 parts per million free, and you seal the tank, and you literally just let it wait, let it sit there, and this can eventually the compound breaks down. You can also treat it with gum arabic nanoprotein products. I've seen that work to a certain extent, and what you're doing is really kind of changing the perceptibility of it at that point. Also, casein can, I've seen it work as well. But really, again, like with all these other defects, the best thing is to prevent this. So if you've never tasted Mousy Taint, there's a good chance you actually can't taste it. Genetically, people have different predispositions to being able to perceive this. And if you do get a Mousy Taint wine at some point, please share it with your friends and your winemaker friends. Not to be diabolical, but to help educate their palate. Because you need to be aware that if you can't perceive it, you need to have somebody on staff that can help you taste your wines and screen out for this. Because I've had a wine that had horrible Mousy Taint, and I had a winemaker right next to me. He said, "Oh, I can't taste it." So we need to be aware of our blind spots, right? So this is where, working with the university or working with your supply partners like myself, we can do sensory tastings for you guys, where we have wines that are spiked, or maybe sample wines that we've found along the way that have these defects, and we can show you what they're all about. So Brett, this is also a big problem in very much the Southwest, Arizona, New Mexico, Texas. There's also a bunch of it on the West Coast. And the compounds that we typically associate with Brett are those horsey, barnyard, and these are sometimes even smoky and leathery. Those are the 4EP and 4EG. There's also an intermediary step, which is 4-vinyl phenol and 4-vinyl glycol. And the interesting thing that occurs is that, I talked about these microbial populations existing together. Pediococcus can actually do one of the intermediary steps to move these compounds from their origin to vinyl phenols, and then Brettanomyces takes vinyl phenols and turns it into ethyl phenols. So this is where typically, if you have a high Brett population, there's a good chance you have a good Pediococcus population. So you can remove these through basically reverse osmosis. I am not aware of an RO unit in the Southwest yet. I think it's eventually going to happen, as some people realize that RO is a very powerful tool. The other thing you can do with reverse osmosis is adjust alcohol. Now, the legality of that, I'm going to leave you to figure out. But there's other things. You can also pull some other wine defects out with reverse osmosis. The thing is, while a lot of West Coast wines are lauded as the pinnacle of winemaking, there's a lot of RO units running around. So it's a powerful tool. Hopefully, at some point, somebody looks into it, because it would really be a good service for you guys' industry. You can use tannins to mask the perception of these, such as we have a tannin called Royal that we found that on a consistent basis, if you have wines that have Brett, it can reduce the perceptibility of these compounds. Also blending, right? So this is, again, changing the matrix, where you take a wine that maybe is very fruity or maybe very oaky, and blend it with one that has the Brett problem, and you can reduce the perceptibility of this. Now, the other thing that maybe the most important thing you need to do before you go blending Bretty wine is to deal with the population, right? So you use something like a chitosan product, like our no Brettanomyces, to actually sequester and damage the cells and cause them to precipitate, and then you rack off or filter off. or filtration. You can also use filtration to remove the Brett population. And then you want to make sure your SO2 is where it needs to be. And then the other thing that needs to happen is if you do have a wine with Brett, if it's in barrel, you need to go through and look at the barrels that have Brett. Sometimes it can be barrel specific, and you need to mark that barrel. Every time you maybe pull samples out of that lot, every time you move this wine that's got Brett or PDO or any of these other microbial populations, you need to really pay attention to your sanitation and try not to cross-contaminate. So this is something that has been on my mind for a long time is how you get a good assessment for the riskiness of doing a cellar operation. So if you have, for example, let's say you're making cider and you just pumped around a wine that's actively fermenting, or a cider that's actively fermenting, and then you just run water through your hose, and then you move a wine with residual sugar. You just inoculated that with an active yeast population. So remember when you work with these wines that you need to follow proper sanitation and good handling techniques. So oxidative defects. Again, this is one of the most common defects that I see out there. In white wines and rosé wines, you get those brown colors. You also get them in reds, but it happens usually at a slower rate. White wines and rosé wines are more sensitive to this. So your rosé should probably be more pink and more red as opposed to orange. Likewise, white wine should be nice and flinty and yellow and not so much brown. So if you do have the development of these brown hues, you can use formulations of PVPP and some caseins to actually pull that color out. In a wine that's got a really bad problem, I know some people will actually do some carbon filtration. I would probably recommend against that in red wines and rosé wines, unless you want to make that wine a white wine, and that carbon can work. Flavor and aroma defects. So this is the acetaldehyde. This is the sherry-like aroma that can happen. This is where you can use some other tools in the toolbox, like tannins. For example, we have one called Tannin Refresh, which kind of refreshes the wine and makes the oxidative characters less perceptible. Also, blending. So you can take, maybe taking a really, really heavy tannic wine and blending it with one that has got some of this acetaldehyde characteristic, and you can mitigate the expression of it and maybe even make a more complex wine. But generally, blending it's useful and you should try it, but be careful that you don't lower the overall wine quality. You want to try to raise the overall wine quality. So if you take good wine and good wine and good wine and mix a bunch of bad wine in it, you've probably got more bad wine than you have good wine. Also oak aging. So what you're doing here, again, is changing the matrix. So you're bringing in a lot of more tannins and aromatic compounds that reduce the perceptibility of the acetaldehyde. And on this note, I'll take a little side street here. It's interesting to hear suppliers and people in the industry call staves and chips and beans and things oak alternatives. They are not oak alternatives. In my opinion, they are barrel alternatives because they're still oak. But anyways, that's just a pet peeve. And it's interesting, and I think it'll become more important because there are oak alternatives that are available from coopers that are from different woods. and the beer industry and the distilled spirits industry is using these very frequently. And if anybody wants information on where to go to get samples of these oak alternatives that are, made from cherry or hickory or sassafras or these other aromatic woods, let me know and I'll be happy to put you together, because that would be it's interesting to say the least. I've played around with it, and it's a good tool in the toolbox, especially when you're talking, you've got a defect. If you can really drive the aromatics in a completely different way, maybe it's something to consider. So, moving on. Filtration and clarification. So, very generally, if you have microbial bloom, like a re-fermentation or a Brett bloom, you got a lot of turbidity, and there's a massive microbial growth. There's three things together that you're going to want to do. You're going to want to remove the microbial population through either fining or filtration. So you're physically removing it. You want to adjust your SO2, and you want to lower the temperature. That's kind of your 1, 2, 3 punch that should be employed as quickly as possible. But from a filtration standpoint, you have depth filtration, which is like plate and frame, pressure leaf, lenticular. You have surface filtration, which would be like a membrane or a cross flow. If you have a lot of microbial growth, you're going to probably want to go through a depth filter first and then into a surface filter, because the surface filters don't have a lot of dirt holding capacity or soil capacity or enough entrapment zones for the microbial populations, whereas the depth filter does. Centrifuge. I don't know of anyone in Texas who has one. They're a very good tool. You can arrest a fermentation without using SO2 and temperature. So, and it will actually separate the microbes from the liquid via centrifugal force. Cold settling. So, if you reduce the ability of these microorganisms to flocculate in the tank, and you sequester them to the bottom by chilling the wine, you can rack off and reduce the population. Micro and ultrafiltration, right? So this is, reverse osmosis, things like that. Spinning cone. This is also kind of the process of clarification and filtration, and it's primarily used for alcohol adjustment. These are just some different examples of filtration. The micro filtration, spinning cone, RO is probably not going to be utilized by most producers. But this is what we're going to try to do to control the spoilage population. I know I'm running over a little bit. I'm going to try to blow through these. Tannins. This is a way to change the perceptibility of an off flavor, protect against further oxidation, and manipulate the mouthfeel to improve the wine balance or change the matrix again. There's also some that combined with proteins. What's interesting about that, this is more of a preventative step. If you have a white wine, for example, that year after year requires a lot of bentonite in order to achieve protein stability, you can look at using something like FT Blanc or FT Blanc Soft or some of these other fermentation tannins to bind up those proteins to reduce your usage of bentonite on the back end. I've kind of already gone over this. So you can do blending. Again, you're changing the perceptibility of a defect or to reduce the concentration. So you'll notice down there towards the bottom of the slide, the solution to pollution is dilution, right? So if you've got, this is one of the primary ways to deal with VA. VA has a legal limit. It's 1.2 grams per liter in white wines and 1.4 in reds. And so, but at that point, you're gonna know it has VA. But not having access to reverse osmosis unit, you may need to blend it out with another wine. If it was me, I'd be at this point wanting to watch my P's and Q's, aka my dollars. You know, if you take wine that you have had this to buy grapes at $2,500 a ton and put all the inputs in, and it's a really nice wine. You know, it's kind of hard for me to want to blend that with the wine that has VA. So maybe not necessarily recommending this, but maybe you want to look at some bulk wine. You know, that is lower price per gallon. That's a good clean wine, and blend that VA out. You know, if you got really froggy and was really trying to just make something that at least could sell, maybe you want to make a sangria with that wine that has IVA. One of the things you do with sangria is probably add quite a bit of water, and so you can dilute that out. So I'm not advocating for one or the other, but it is a possibility. So you can blend to reduce or raise the pH. I think in Texas, most likely it's going to be a necessity to reduce pH, not raise it. You could also blend in something like Muscat or Riesling or something that's a Gewürztraminer that's pretty aromatic. And you can reduce the perception of an undesirable aroma for example, maybe like a little Brett. If you could just take those aromatics and drive them more into a floral area and maybe cover up some of the horsey aroma, you might actually make a better wine, more complex. And then you can blend to modify color. So this is if you have some wine that's brown, you know, taking wine with a larger phenolic content and larger tannin content and blending it in can help dissipate that color and improve it. You know, the other thing that you may be able to use is some of the concentrated products that are out there, meaning, you know, mega purple or mega red or, you know, whatever it is. Could also be just straight concentrate. And again, I'm not trying to promote this necessarily, because I know some people might be very much against buying a drum of Petite Sirah concentrate from California Concentrates and blending it in, but it is something that you know, is a tool in the toolbox. So if you want more information on that, let me know at the end of the presentation. So if you have a defect and it's microbially based, we need to do something about it. And we talked about kind of the big three, which is you know, chilling the wine down, filtering it, and getting SO2 into it. But I do want to talk about SO2. Even at a professional level that a lot of us are operating at, there's still some misconceptions about SO2. So the chart on the right hand of the screen correlates the free SO2 that you need at a given pH to achieve proper molecular SO2. So at the top it says 0.8 ppm molecular SO2 and 0.5. Generally speaking, very broadly, you're making a white wine. Winemakers have typically said 0.8 is where you want to be. Red wine's 0.5. I like 0.8, especially if you can get there based on your pH. But if you notice, if you're in the 3.8, 3.9, 4 range, look at the amount of free SO2 you need. Unfortunately, that is significantly higher than the sensory perception threshold, and the wine's going to smell like SO2. So, you know, this is where acidulation comes into play. And then if you don't want to acidulate, you need to probably do some things using some stability agents other than SO2, and we'll talk about those in a minute. So SO2 is a microbial inhibitor. It does not kill anything. And I repeated that, and I put it in red and in all caps, and so I'm yelling, because I hear it a lot, and it doesn't kill things. It does inhibit microbial populations to an extent. But for example, there are spoilage yeasts that have extracellular sulfite binding compounds. So what they do is they actually produce a compound, secrete it across the cell membrane, and it binds with free SO2, thereby lowering the SO2 down to a point where they can have a nice little microbial party. So if everything else that we've talked about hasn't kept you up at night, just know that there are little bugs that can do that. And I wish I could tell you if it's schizo or zygosaccharomyces, but it's one of the two. I can't remember off the top of my head. Anyways, so SO2, depending on your pH, your initial population, and your dissolved oxygen, temperature, and ethanol, these things work in synergy. And I'll describe the situation. So low dissolved oxygen and relatively low temperatures with elevated alcohol levels and low pHs is a really nice situation to prevent the growth of spoilage microorganisms. You know, there is some magic number around 12.4 to 12.6% alcohol that you get a lot more stability. So if you're looking at some of the products that are out on the market and you say, hey, I want to make a low alcohol wine, I want to make a 9% wine, which you may want to do. There's a lot of market for that. Just know that your standard practices that you've been using on your 14% wines, you're in a different territory at that point. And you can use the same amount of SO2, sorbate, in your standard filtration regime, and at 7-8% alcohol, you can have the growth of a lot of spoilage microorganisms that you wouldn't at, like, let's say 12.7. So anyways, I think that's enough about that. We're going to get into some other stability agents. So one of the really cool things that's come out in the market the last couple of years, and a lot of the vendors have them, are these chitin glucan or chitosan products. We have two different formulations, no Brettanomyces and Bacillus. What's nice about it, the no Brettanomyces is fairly specific to Brettanomyces. Bacillus is specific against bacteria. What's nice about that is it's effective on gram-positive and gram-negative bacteria. So this is your lactic acid bacteria as well as your gluconobacter and your acetobacter. So it's broad spectrum. And these are very powerful tools. They're non-allergenic. They're derived from Aspergillus. And so when you have these high pHs and you don't want to add 100 parts per million free SO2, you can use these products to reduce the microbial populations. And so this can be very effective. Now, sorbate, this is very common. It's kind of something we pulled from yesteryear and we're still using it. You have to use it in combination with SO2. And it's good at preventing re-fermentation by Saccharomyces. So you'd use this in maybe residual sugar wines. It is not effective on bacteria. In fact, it's actually a food source or a carbon source for certain bacterial populations. And this is where you get geranium taint. If you've ever smelled geranium taint in a wine, you know that the winemaker had an oops and didn't control their bacterial populations. And also, it has a very unique sensory property to it that it eventually breaks down to ethyl sorbate. And you can get notes of pineapple or celery, which is exactly what I want in my wine. Maybe pineapple. I don't know about a celery stick. I like them in my Bloody Mary stuff. Anyways, then we have things like Velcorin. Scott Labs is the exclusive distributor in the alcohol industry for Velcorin. It's a very unique product. Has unique handling characteristics that you must abide by, but it is very effective against yeast and moderately effective on bacteria. Again, all these things are working in synergy, right? So pH, alcohol level, microbial population, all these factor into how effective these stability agents are. It does require special dosing equipment and special handling protocols. One industry that's using this extensively is the cider industry because of their low alcohols and relatively high pHs. Everybody that's tuned into this webinar has had a product that's been dosed with Velcorin because it's used in tea drinks and sports drinks like Gatorade and Powerade and things like that. So we've all had it, but some winemakers have philosophical disagreement with it, and I can certainly understand that. But it is nice in certain circumstances. And we can talk more about that at a later date if you'd like to learn more about Velcorin. I'd be happy to talk about that. Temperature control. So we can either chill our wine down, like we've talked about earlier in this presentation. Useful to limit the growth of the microorganisms. Also encourage the formation of tartrates, and you can actually get somewhat of a fining effect with when you form tartrates. Causes things to start to flocculate downward in the tank. Also, it will slow reaction speeds of certain chemical processes. One thing I want to be very careful with here is saying you should chill all your wine down really, really cold. The reason I want to be careful with that is you greatly increase your gas solubility, in particular dissolved oxygen. When you, for example, take a white wine and chill it down for a very long time to try to achieve tartrate stability, you can dissolve a lot of dissolved oxygen. And then the wine, when you rack it off and you warm it back up, the wine goes brown and chews up a lot of your SO2. So, we are all familiar with bottle shock, which is primarily exposure to oxygen. I think there's also barrel shock. When you take your wine out of tank and go to barrel, I think you have barrel shock as well. And then I think there's chill shock. So after things come out of chilling, cold stability, the dissolved oxygen binds up the SO2 and you have browning. And you can also have the growth of spoilage microorganisms at that point too, because you don't have good SO2. So heating. This is another possibility. So this would be a powerful tool. Let's say, for example, there's a flash unit in Texas now. If you have moldy fruit, which is you're probably going to have all sorts of mold and fungus and yeast and bacteria, all sorts of things growing, you can put it through the flash system and help mitigate the growth of those organisms. We all are familiar with pasteurization. That works. I'm not a huge, sorry about that. Pop up on my screen here. You probably don't want to use pasteurization on your wine, but people do use it on cider. So it is a possibility. Thermovinification is kind of an interplay of the flash to taunt to a certain extent. And you're probably saying, like, Luke, why did you say that pasteurization, flash to taunt can help kill these microorganisms, but then you say right below, well, it can increase microbial growth and reaction speeds. And I'm saying I want to delineate between the levels of temperature. So pasteurization, you're talking 140 to 200 degrees Fahrenheit. If you wanted to increase microbial growth, warm things up to about 75 degrees. They're going to proliferate a lot at that point. So you take them up to 75, they're really happy. Take them up to 185, and they're dead. So this is the same for humans. So at 11 degrees Fahrenheit, I am in by the fire. At 65 degrees, I'm out hiking around, and at 120 degrees, Central Valley of California in the summertime, I'm near the AC, and I think we all would be. So think of it that way. So again, I want to also, we touched on this briefly before, but specific inactivated yeast, powerful tool with a lot of numerous benefits. You can get mouthful improvements. You can, again, reduce the perceptibility of certain compounds. It's a very soft method. It's it's being very nuanced with your additives instead of something like carbon that's very aggressive. I've seen rescue or SIY like that reduce residual copper. So if you did use copper to bind up with volatile sulfur compounds, but you've got too much and it's above the legal limit, you can use rescue to drop that back down. It's really pretty awesome. You can also remove inhibitory toxins. So like, let's say you have the stuck fermentation and there's some fatty acids that are causing some problems for your restart. You can use rescue or something like that to absorb those compounds, rack off, and then go ahead and re-inoculate. You'll be rocking along your way. So they're very handy, and they're just autolyzed yeast, and they can have a lot of other benefits as well, like bringing out some proteins and some umami flavors, and also potentially help you in tartrate stability. Inert gases, real quick. You got nitrogen on the left. You've got carbon dioxide or dry ice in the middle, and argon on the right. So we'll get into this a little bit. So generally, this is curative. This is treatment. This is handling in your cellar. So you use these in different areas to protect against oxidation, to limit the growth of certain oxidative spoilage microorganisms, but they have different properties. So real quick, argon, it's great for inerting headspaces, has very low solubility. So you're not going to get argon bubbles in your wine. It's heavier than air, so it does have a little bit more of that protective layer over the top of the wine, but it is very expensive. So I would say you want to use sparingly. Carbon dioxide, again, great for inerting headspaces. It is high solubility, so you can get some effervescence. So you want to be careful with that. It's heavier than air, so again, you do get that protective layer, although it is not this perfect blanket that we think it is. There is some mixing in the headspace, so you still can have some oxygen integration. The good news, it's inexpensive, it's easy to add. You can use dry ice, you can get blocks of dry ice or cubes of dry ice, or you can use gas. Nitrogen, you can actually, because it has very low solubility and it's inexpensive, you can use it to remove the other dissolved gases such as CO2. And maybe more importantly, you can use it to literally drive out dissolved oxygen. And if you want to talk about that later, I would love to talk to anybody about how to use this. So moving on, I know we're covering a lot, but we're going to keep after it. I don't think I have too many more slides. pH manipulation. So I'm a big believer in acidulation, especially at the juice stage. Real quick, if you're doing a red wine that you're going to put through malolactic, you're probably going to want to use primarily tartar to adjust the pH. If it's a white wine or a rosé wine that you're not putting through malolactic, I like to do like a 2/3 tartar, 1/3 malic blend. You could use citric acid as well, but it has kind of that citrusy flavor component to it, and it's also a carbon source for certain bacteria, so be careful with that. They have different strengths, so different with their conjugate bases, but some of them are stronger than others. So you want to do bench trials. You can't use them interchangeably. What's nice about it, especially at the juice stage, and really at any stage, you can use it to drop the pH and really shock the microbial population. So if you've got some fruit coming in from the high plains, it's high pH. There's been some microbial shenanigans, and you drop the pH down to 3.62, which is a really nice little number. You can really shock those bad microbes into submission, and also you get increased effectiveness of SO2 and other stability agents at lower pHs. Deacidification, I don't think it has much role in Texas, but it is a possibility, and most of the time we have high pHs, not low pHs. But some people do use it. Below about 3.2, yeast start becoming inhibited. And so if you have something that's really low for some reason, you may want to think about raising the pH. So ion exchange, you can also, you're exchanging the ions and adjusting your potassium levels, and you can really get some pretty good effects, but it's very expensive, uses a lot of water, and it's probably not feasible for most people unless, you know, everyone banded together and decided to get an ion exchange unit. And you can see the little table on the right on what you can do versus chilling contact. Because your other way to adjust your pH is through traditional cold stability, depending on your pH. So above 365, your pH goes up. During cold stability, below 365, it goes down. So be careful with doing that. And we could probably do a whole seminar on that as well. So this is, the next slide is saying like, hey, if you don't have a chiller and maybe you have a wine that's 3.7 pH and you don't want to do traditional cold stability, or you're worried about the dissolved oxygen pickup, you can use these things called crystal inhibitors. So this is going to prevent the precipitation of potassium bitartrate, those little wine diamonds that some people like to call them. So CMC, carboxymethoxycellulose, I probably messed that one up, but really mostly used in white wines because it can have some negative color effects. It can exacerbate protein instabilities. It can be very difficult to add. I've had people say that they never want to use it again, but it's fairly inexpensive, and you can't use it with wines treated with lysozyme. And so any one of these that I'm going to mention today as crystal inhibitors, I highly recommend sending them to your supplier to have them run trials on these crystal inhibitors to see how effective it is, because your potassium level and your calcium level and colloidal matrix, all these things will play into how effective these compounds are. So one that we have is a manoprotein based. It's called Claristar. You can use it reds, rosés, and whites. It has much less effect on protein instabilities, but it can still be a possibility. So you want to be careful with that. And it must be in the final blend state. So you can't add it and then change things up and expect the cold stability that you did achieve to carry on. Neither effective against calcium bitartrate, FYI. So if you do have calcium bitartrate precipitation in the bottle, these are not going to do anything for you. There's a new product coming on the market called potassium polyaspartate, KPA, sold by a competitor of ours. It doesn't have some of the eccentricities that these others do. Again, bench trials are recommended, and they can help you with that. I think Enardis is the one that has it. I don't know if anybody else is coming online with a product, but some of the parameters that CMC and Claristar need are not necessary for the KPA. So it's seemingly a pretty good tool. It's not legal to use yet, so there's some waters to be navigated with the TTB there. But it is coming on the product. I think that the name is Zenith, what they're selling it as. So that's something to be aware of in your toolbox. So I think this is the last slide. So what other things can we use? Well, sometimes a defect can be way too much SO2. Let's say you wanted to add 4 grams and they saw 4 pounds and they added 4 pounds. What are you going to do? These kind of things do happen every once in a while. Hydrogen peroxide, H2O2, can be used to reduce SO2. I would recommend looking at the legality of that, depending on your state, depending on your federal, and then if you plan to export at all. It's also a super strong oxidizer, so be careful because you can take a small problem and turn it into a really big problem. But Last thing I really want to talk about is a curative fermentation. So, for example, if you have a wine that is really oxidized, you could potentially put it through a secondary Saccharomyces fermentation using a champagne strain, and maybe clean that up significantly. And people do it all the time and they make champagne. So that's one of the reasons why when you're out and about and you want a nice clean glass of wine, most of the time a method champenoise wine is a very safe choice at your local Chili's or wherever you're at. So I think that's about it. There we are. Any questions?
Thank you very much, Luke. Please, if you have any questions, type them in the Q&A box on the screen there, and we'll try to answer them all.